Hello everyone, thank you for joining us. My name is Nicole McCune and I'll be starting off our presentation on identifying rare blood type mutations through personal genome analysis. I'm presenting today alongside my group members, Manit, Zoe, and Skyla. Our project focuses on whether direct-to-consumer genetic testing, specifically the 23andMe V5 SNP array, can reliably detect rare blood group mutations. Blood group genetics is incredibly complex, with over 1,500 alleles across more than 43 blood group systems. Some of the rarest and most clinically important phenotypes, like Bombay, junior negative, KID null, and RHD variants, often required specialized sequencing and serologics testing to identify. To explore this, we compared known rare blood group variants from ClinVar to the SNPs actually included in a real 23andMe raw dataset. We looked for true allele matches to see whether these rare phenotypes could be detected. What we found is that 23andMe covers only a very small number of blood group related SNPs, about 6 to 8 in total, with only 2 clinically meaningful markers. As a result, most rare blood group mutations simply can't be identified using consumer genomics. Overall, our study highlights a significant gap between personal genetic testing and the needs of transfusion medicine, and points to the importance of genome sequencing for accurate rare blood type analysis. With that overview, I'll now pass it on to the next presenter.
Okay, so blood types aren't just simple labels like A, B, or O. They're determined by specific genes that control which antigens appear on the surface of our red blood cells. The three major systems are especially important: ABO, RHD, and KEL. The ABO gene decides whether we produce A or B enzymes, while the RHD gene produces the RHD antigen, which is highly immunogenic and clinically significant. The KEL gene adds another layer of complexity since KEL antigens are among the strongest triggers of immune reactions after ABO and RHD. These genetic differences matter because variations in RHD and KEL can lead to serious outcomes like hemolytic disease of the fetus and newborn or transfusion complications. Thanks to modern genomics, tools like SNP analysis and next generation sequencing now allow us to detect rare variants in these genes, even from consumer generated data. Our project builds on this approach inspired by the real world case of James Harrison to explore how personal genome data can reveal uncommon blood type mutations with meaningful medical impact. Thank you.
In this project, we compare ClinVar blood group variant with raw 23andMe data to see whether rare blood type mutation can be detected on 23andMe SNP chip. We filter ClinVar to rare blood group genes and process the 23andMe file with about 650,000 SNPs and matches both dataset by chromosome and position. A variant was only counted if the genotype included the ClinVar alternate allele. ClinVar showed the 100 of variants across several rare blood group genes. These genes are associated with phenotype like Bombay, Bombay junior negative Duffy null, KID null, adult I, P1, PK, and different RH types. When we compare this variant to the 23andMe dataset, only a very small number of position overlapped and none of the rare blood group phenotypes were detectable. The only trait the SNP chip could reliably capture were common ones such as FUT2 secretor status, ACKR, Duffy status, and a rough ABO estimate. Overall, this highlights that the 23andMe array provides a very small coverage for rare blood group genetics, so we can basically use this for a purpose we're looking for.
We set out to test whether 23andMe's SNP array could detect rare blood group phenotypes. What we discovered is that while the array offers some insight into common variants like FUT2, which determines secretor status, and the ACKR1 or Duffy null variant, it's not built for detecting rare clinically significant blood types. This consumer array only looks at a limited number of SNPs, so key alleles for rare phenotypes such as Bombay, junior negative, adult I, KID null, RHD variants, P1, PK, and para-Bombay aren't included. This means that even though 23andMe's data can be interesting for personal traits or ancestry, it cannot provide meaningful information for transfusion medicine or rare donor identification. Our findings highlighted the gap between consumer genomics and medical grade genetic testing. To reliably detect rare blood group mutations, we would need more comprehensive tools like whole genome sequencing, targeted blood group panels, or even traditional serologic testing. Overall, this project really emphasized how both the potential and the limitations of consumer genomics. It shows how these tools can raise awareness of personal genetics, but also reminds us that for clinically significant applications, a more comprehensive sequencing method is essential. Thank you.